Thanks for the introduction, Brian, and welcome everyone. Good morning, good afternoon. I think it's the virtual world, so we're all coming in from everywhere, which is great. And so excited to be here, and excited for the conversations as Brian highlighted. And I'm excited to welcome Po to the stage here with me as well for a conversation I think is on everybody's mind on a regular basis when it comes to cell and gene therapies. So Po, before we jump into the conversation today, why don't you give you an opportunity to give a quick background on yourself, and how you find yourself where you are today.
Yeah, thanks, Dominic. Hi, everyone. My name is Po, Yeh-Chuin Po, but I go by my last name, Po. I am currently the Senior Director at Beam Therapeutics. Beam is a company that focuses on base editing using a modified CRISPR-Cas9 system to do single base pair edits. And I am currently also the CMC lead for the immuno-oncology franchise, so primarily focus on the CAR-T, CAR-NK programs. Prior to that, I've also had some experience with hematopoietic stem cells, CD34 stem cells, and then prior to that, it was pluripotent stem cells, differentiating them into pancreatic beta cells. So about a decade in the biotech industry and primarily focused in cell and gene therapy.
That's awesome. So obviously a little bit of overlap on the topic we have today, right? So, as we know, we're here to talk about CMC and process analytics for cell and gene therapies. When I think about CMC challenges and the challenges associated with CMC and cell and gene therapies, these have been discussed and debated probably at least I would say for the past decade. It seems like we've been talking a lot about this same issues for quite some time. So we all know about the impact around the regulatory success of these products, yet many companies continue to fail to get it right, say the first time. So with that in mind and thinking about overcoming cell therapy and cell and gene therapy CMC challenges, let's jump right in, Po. So thinking about this and where you've been involved, your experience balancing continuous process innovation and then with the FDA, right? Because we know where we are. We're an immature, developing industry, and innovation is running at a speed that's perhaps quicker than the FDA and the regulatory bodies can keep up with. So, from your standpoint and your experience, how have you found that, and how do you manage that as a company?
Yeah, no, thanks for the question. And I think it's something that many people really need to start thinking about, not just in the near term, but also in the long term, right? There's a phrase that many people use, phase appropriateness. What exactly is the phase appropriate strategy when it comes to your CMC strategy? We all have to understand that as we actually have a CMC strategy, we have to keep that big picture in mind, where it's not just the immediate needs at which you're trying to address, but further down the line, what exactly is it that you're lining yourself up in order to mitigate or to be able to accelerate your program faster as you kind of progress through the life cycle of the program. So one of the things is really managing the improvements to your manufacturing processes, right? So take, for example, if you were to have a cell therapy product, you develop a process for manufacturing and scaling up. But many a times, whenever you scale up, your initial scale up manufacturing process is appropriate for the phase 1 clinical trial. And it's not always the same process at which you use when you get to pivotal or commercial. So making sure at least having in mind what exactly is it that you're hoping to scale to, what is the target number of cells, what exactly is it that you're hoping to accomplish when you get to pivotal and commercial, really helps you to formulate what kind of improvements are there that needs to be put in place, right? So take, for example, if you were to think about some of the critical materials like guide RNA, mRNA, lentiviral vectors, what are the improvements to the manufacturing processes that need to happen in order for you to get to your pivotal and commercial readiness. So at the early stage of the company, you might actually say, hey, this is sufficient for us to get to phase one. But what about the process improvements for you to get to phase two and beyond? Those are the things that you really need to start thinking about, especially as you interact with the FDA. Right? And
Yeah, go ahead. Sorry, go ahead.
no, I was going to say that, the FDA interactions is also something that you have to be strategic about, because many a times you don't just go straight in with an IND amendment to your improved processes. You usually have a type C meeting, and especially in those interactions, what kind of specific feedback are you trying to get in order for you to get your process improvements from one place to another, from one generation to another, right? And then really also thinking about, hey, as far as your scale up or scale out strategy, how do you actually get your manufacturing process to a scale at which it's pivotal and commercial ready? So those overarching thought process is very critical even in the early stage of your manufacturing process. Your CMC strategy in the early time point of your life cycle is just as critical as the CMC strategy later on in the life cycle.
Well, now let's just piece that out a little bit. So I think this is going to come up regularly. The balance, right? So we're always trying to strike the right balance, and doesn't how do you do that when you're trying to? Oftentimes, a company is trying to quickly get to the clinic, right? I mean, it's all
Yeah.
about speed for the most part, getting there. So how do you balance that out? And with your process, your CMC requirements, to then separate that obviously from the next phase, right? Because you're often two different mindsets, getting to the clinic, and then eventually you're going to think about going beyond into the next later phases. So how do you work that out especially if you're going into your early stage and you're thinking through that? How do you not make the mistakes?
Well, I can tell you with the many programs that I've been involved in, there's never one program that is perfect. Mistakes always happens, right? And that is the nature of the industry that we are in, right? You always have deviations, you always have different investigations and putting CAPAs in place and so on. So continuous improvement is something that is natural for this industry. But nonetheless, we still need to have a CMC strategy, right? It doesn't mean that we just go in and fill ourselves out. We need to have a proper CMC strategy. So when we talk about CMC strategy for the early phase versus the late phase, we need to talk about the phase appropriateness. So meaning that in your early phase of your clinical trial, let's say in phase one, what exactly is the target number of patients that you're trying to get to? From there, you can back calculate as far as, okay, how many cells do I actually need to produce in this phase one? In an allogeneic setting, you will be thinking about, okay, how many batches do I need to generate? In an autologous setting, okay, how many runs do I need to generate? How many manufacturing campaigns do I need to meet the number of patients that I'm hoping to get to in this phase one? So the skill at which you're trying to get to, especially in an allogenic setting, the skill at which you're trying to get to in a phase one strategy is usually much lower than that of a later stage. And that is totally fine. And what you need to start thinking about is, how do you therefore show the improved processes later on is comparable to that of the current process which you're using? And making sure that you have that comparability strategy even earlier on in your phase one trial is very critical, because you don't want to come to a point at which you're already in your phase one and then you realize that, oh, we have to make improved processes and we didn't really think about, okay, how do we actually show comparability? That becomes an issue. So your strategy upfront needs to inform your strategy later on, and having that broader picture, not just in the next 2 3 years, but 7 8 9 10 years, that strategy really helps you kind of carve out your CMC strategy here.
Yeah, I imagine, as with anybody, it's balancing the short term and thinking about the long term at the same time. So you know, perfect timing here in a sense of, when you think about that, and you touched on it already, but implementing that proper comparability strategy.
Yeah.
And, from a manufacturing perspective, also, I wonder if we touch on, I don't know if you've had experience when it comes to either doing this and then transitioning this, say, to an outsourced manufacturing partner too. How do you balance that? How do you work that in from a comparability strategy from
Yeah.
your own set, from your mindset, then also working with a partner to build out that manufacturing?
Yeah, and this is another kind of important thing, right? When you're selecting your CMO or when you're selecting your manufacturing partner, it's important to start thinking further down the line because there are many manufacturing facilities that are ready for the early phase one clinical manufacturing, but they are not really ready for commercial manufacturing. So making sure that you're picking the right partner, and it's totally fine if you were to pick a CMO that is appropriate for your phase one trial, and then after that, switching your manufacturing facility from one to another in and of itself requires comparability data packages. You need to file an IND amendment for that. So, of course, ideally, you want to be able to pick someone that you can actually run with for the next 20 years, but that's not always the case. So, identifying the need that you have right now and really thinking down the line, okay, what is the comparability data package if I were to switch vendors, the manufacturing sites? Right? And it's not just the manufacturing sites often that actually changes. You actually often change your manufacturing process as well. So if your phase one clinical trial manufacturing is actually with this CMO, you're then switching for your pivotal and commercial to a different CMO. Often it is you also implement process improvements. So the important thing is really to understand what exactly are the changes, perform a risk assessment before you actually execute the comparability study design. And this has to be a very well thought out process because the FDA will ask a lot of questions and probe very detailed questions, right? And I can even go into further details that it's not just that strategy, but the details such as your analytical comparability, things such as your safety, your identity, your potency readouts, those are needed for your process improvements as well. So the critical part that really comes into play is that as you strategize and make sure that you have proper comparability strategies, you're also thinking about how you have to retain certain drug product vials in your Gen 1 manufacturing process, such that you can then kind of analyze it later on side by side with the future generation of your manufacturing process or the new manufacturing site that you're doing it at.
But I'm sure there's plenty, but is there, as you're thinking about the the comparability strategy and doing that risk-based assessment, are there a couple of key components in there that are just seemingly obvious when you're looking at this? You mentioned some of the analytical, aspects, and
Yeah.
luckily we're going to have another discussion here shortly that we can go into deeper detail, so I don't have to probe you for that and put you on the spot. But just thinking about when you think about a proper comparability strategy, maybe there is, maybe it's not obviously a one size fits all, but I
Yeah.
wonder from your experience
Yeah.
where
And
you
I'm sure,
found that.
yeah, I'm sure Bruce can talk a lot more about this after this. He's the expert in that area. But yeah, there are several analytical aspects that needs to be really taken into consideration, right? The first that I would like to mention is take for example in a CAR T program. Uh, Many a times people use interferon gamma as a potency release. When you actually get to pivotal, that might actually not be sufficient. You may actually need to have something that measures the mechanism of action, right? Or something like that. So many a times, in fact, not many a times, I would say always, you would actually have analytical improvements as you move from one stage of your clinical trial to a different stage, different phase of your clinical trial. And when you start thinking about how you improve your analytical processes, you're also trying to implement in parallel the process improvements or the site change, the site manufacturing, the manufacturing site changes. You're kind of implementing a bunch of different changes as you're implementing your analytical change at the same time. So how then do you actually make sure that you are able to show comparability to your first clinical manufacturing drug product? And that's the part where the importance of retaining drug product vials, especially in an allogenic program, for characterization, for comparability, those are extremely critical. And we have seen in many trials, and some folks here would have been familiar with that as well, that there have been trials where you actually get to a stage at which you're implementing changes and you don't have enough retains to compare it with your previous phase one clinical trial. And a lot of those data is basically wasted. You can't really bridge it.
Well, so perhaps that was it right there, right? The retain vial, having those, I've seen that in the past too. It's hard to think about 5 years ago,
Yeah.
holding on to samples and then maintaining, traceability of those samples and information, which we'll talk about it separately because we don't want to get into that now. But you know, question then, it's obvious it's here. So not so much what are they, but when? When in your mind do we make pivotal changes for the product? And again, that could be, I'm sure it's an open-ended question
Yeah.
and answer.
Yeah, and before we even move to when, just want to mention as well that the retain vials that we were talking about, it's not just a drug product, right? There is also the starting material. So if you were to actually think of many of the trials that are out there, especially in allogenic programs, you start with healthy donor apheresis. That starting material vials of those needs to be retained as well because it's so important as you go down the later stage of the clinical trials. And as far as when to make these changes, this is something that is very dynamic, right? It really depends on the program. It really depends on the company, where you're at, where you hope to be. But this is where Type C meetings is very critical. It's very important. Type C meetings with the FDA. Uh, those Type C meetings is where you actually come with a clear comparability strategy, where you actually bring that strategy to the FDA and you get them to buy in on it and you get their feedback. And very seldom do they actually say, hey, perfect, everything good to go, and just execute. They always have clear feedback to you, making sure that you have clear and pointed specific questions during that type C interactions will help you define what your comparability data package needs to be. Right. So take for example, when you actually start thinking about where you are in the clinical trial, depending on where you are, the number of ends that you need to generate in order to show comparability may significantly differ. So if you are still early on, preclinical, you haven't even dosed a single patient, it's much easier to implement some of those
Right.
changes and show comparability there. Whereas if you're already in your pivotal studies, and you're trying to make process improvements, you're trying to make changes during your pivotal trial, you may actually require a lot more to show comparability there. So it's really a regulatory and clinical strategy needed to identify the timing of when the CMC changes should be implemented during the clinical life cycle.
So much like anything, constant communication and certainly cross-functional collaboration, say with obviously internal groups, but then your manufacturing partners, the regulatory bodies. So, I want to touch on one other point here too, and probably the last point in as we transition. But when you think about where we are and you think about making changes, we'll bring up the beautiful topic of standardization. So again, knowing where we are today and what you've seen, what you've experienced, is are there areas today that we can consider, standardizing, so from your standpoint, what's possible today versus, and more importantly, perhaps what's really appropriate, because you don't want to standardize something perhaps and then have to make a change.
Yeah, and standardization is a critical point, right? Platform and standardization. You can do it internally within your company, within your organization, or you can do it with the broader industry folks, right, with your peers. Talking first about your own company, your own organization. Many a times people try to do things very quickly where they just want to nip it in the bud and they end up doing it one program at a time instead of just trying to approach it from a platform uh, approach where you actually say, hey, what is it that we can leverage across all this multiple critical materials or drug substances? What is it that we can actually approach it as a platform instead of doing it individually? Instead of filing multiple IND amendments, can we actually take it as a platform and have one single IND amendment that can be applied across multiple different programs? So one example that I can think of is take, for example, those companies that use CRISPR-Cas9, right? You need a guide RNA. Uh, when you actually have improvements to your guide RNA manufacturing process, often it's not just one specific program that has improvements on. You want to leverage that improvement, that manufacturing process improvement across all the programs, all the different guide RNAs that you're trying to manufacture for the different drug or different disease types that you're trying to target. So when you have multiple guides with not multiple INDs, can you take it as a platform approach where when you're actually filing an amendment or if you're actually improving the manufacturing process of the guide, can you make it a platform approach where, hey, the guide sequence might differ by only uh, 20 base pairs or 20 nucleotides, but significant amount of that sequence is actually similar across board. So can we leverage a platform approach where, hey, this process improvement or this process standardization can therefore be applied across multiple programs? And then as you take a step back, now if we were to think of the industry as a whole,
Right.
what kind of process standardization is possible and what is appropriate? Now you start thinking about, hey, there are some things that we could potentially do even as an industry, or maybe a consortium can be formed around this, whereby we actually start thinking about, hey, many of the cell therapy programs are moving towards an allogenic setting. When you actually get to an allergenic setting, there are so many things right now that every company is using by themselves and it's not really standardized. Can we come to a point at which the industry is standardized across, hey, uh, when it comes to 15-liter bioreactors, we should standardize using Masterflex tubing. Uh, and then we can have a drug master file for a specific tubing set that we're using or something like that. Therefore, we can then just cite it or reference it in the amendment that we actually have. That will be tremendously helpful for many of the different companies that are working on cell therapy there.
Yeah, I think you make an excellent point and distinction. I was speaking broadly, but as you said, there's the company and your own platform and what you can standardize and when versus the industry. And I think both of those go hand in hand. But as you touched on, there's definitely a couple of areas within an industry as a whole that I guess if there's one aspect you'd like to see standardized from an industry standpoint that isn't today, what's on your short list?
Oh, there's
What
several.
we can achieve.
I I I have a pet peeve on tubings. I really feel like the tubing set that we are all using right now is kind of grandfathered in from the blood industry. Everyone's using PVC, all the instruments such as the Prodigy and things like that, they're all kind of PVC tubings, but it doesn't always work for every single program. If we can actually standardize, especially when we actually get to a larger scale, hey, maybe we can actually use a different set of tubings instead of always just using the PVC tubings. That would be something that I would love to see as an industry.
Plastics brings me back to my earlier days as well. So I feel your pain and challenge there. So, well, obviously this has been fantastic, Po, and thank you for your insights, and your expertise is certainly welcomed, not just in this forum, but across the industry. So hopefully I'll remind the audience here that if you have any questions for Po or any of the speakers, please remain on for the, when we're finished up with the conversations here. And Po, thank you for your time, and we'll look forward to connecting back with you at the networking session.
Thank you, Dominic. Pleasure to be here.